Hello, and welcome to today's Fluidigm FAS Friday session. My name is Andrew Draghi, a North American mass cytometry field application scientist based here in Connecticut. I'm here today with my colleague Wendell Smith, who is the senior field application scientist specialized in proteomics and resides in Southern California. We'll be taking your questions in about 10 minutes or so. Please enter your questions into the question box at any time during the presentation, and we will answer them during the Q&A session. In the meantime, we will start by highlighting the use of mass cytometry and imaging mass cytometry in checkpoint inhibitor studies. Profiling the human immune system is a key application used in translational and clinical research to look for biomarkers of disease or response to therapy. It is essential in driving the search for more effective treatments for almost any disease, including cancer, autoimmune diseases, or infectious disease. Fluidigm is well aware of many analytic challenges faced when studying immune-mediated diseases and the complexity of the immune response in health and disease. Challenges for these types of studies can include limited and precious samples, longitudinal data comparisons, cost-effective and efficient analysis, incorporating new findings into ongoing studies, and the ability to find the unexpected. Mass cytometry and imaging mass cytometry can address these challenges through the use of high-dimensional, comprehensive panels, which allow the most information to be derived from precious samples. The use of mass cytometry to study the effects of immunotherapies targeting checkpoint inhibitors like CTLA4 and PD1 have been on the rise, providing insights into the mechanisms involved in generating anti-tumor responses. Mass cytometric profiling and characterization can be performed on cells isolated from peripheral blood or the tumor itself, either at baseline or during the course of treatment, to help uncover novel or rare cell subsets, define biomarkers or immune signatures relating to drug response, stratify patients into certain risk groups, as well as potentially help predict clinical response to therapy. Let's take a look at some papers that investigate the immune response to checkpoint inhibitor therapies using mass cytometry and imaging mass cytometry. In this paper from Jim Allison's lab at MD Anderson Cancer Center, mass cytometry enabled comprehensive characterization of checkpoint blockade mechanisms in mouse tumor models and also human melanomas. They found that anti-PD1 and anti-CTLA4 utilize distinct cellular mechanisms. Both anti-PD1 and anti-CTLA4 target subsets of exhausted-like CD8 T cells. However, CTLA4 blockade can also induce expansion of ICOS-positive Th1-like CD4 T cells. Lastly, the authors also found that T cell responses to different tumor models are fundamentally similar. In another paper from the Allison group published in 2019, distinct cellular responses to combination anti-CTLA4 plus anti-PD1 checkpoint blockade were identified. Mass cytometry was chosen as the sole method to investigate cellular responses to either dual immunotherapy or monotherapy. Murine tumors and peripheral blood from melanoma patients were assessed with 40+ marker CyTOF panels. Metaclustering identified 14 tumor infiltrating T cell subsets differing in frequency between both mono and dual immunotherapy conditions. Combination therapy and anti-CTLA4 therapy, but not anti-PD1 therapy, were found to elicit a switch from expansion of exhausted CD8 T cells to that of an expansion of activated effector CD8 T cells. In this paper from Buchard Beecher's lab at the University of Zurich, 3 panels consisting of 30 plus markers enabled in-depth characterization of immune cells from whole blood of patients with metastatic melanoma before and after anti-PD-1 immunotherapy. The authors identified immune cell subsets that could be predictive of response to immunotherapy. In particular, the frequency of classical CD14 positive, CD16 negative monocytes with migration and activation marker expression may be a strong predictor of response to therapy. Additionally, T cell activation status and frequency of T cell subpopulations were also altered in responders during anti-PD-1 treatment. In this paper by John Ware's lab at the University of Pennsylvania, they used blood-based immune profiling to reveal pharmacodynamic changes in the response to anti-PD-1 therapy in patients with advanced melanoma. The clinical response correlated to the ratio of the reinvigorated exhausted T cells to that of tumor burden. The identification of these rare circulating exhausted T cells in the blood may therefore help predict clinical response to anti-PD-1 therapy in the future. This paper from Evan Noel's lab, where he was at the Singapore Immunology Network, highlights the use of mass cytometry to characterize neoantigen-specific CD8 T cells in tumor-bearing mice following checkpoint blockade with anti-CTLA4 or anti-PD-1. A broad range of predicted candidate tumor antigens was comprehensively screened using CyTOF approach with multiplex tetramer staining across multiple tissues. They showed that immunotherapy can alter the frequency and the phenotype of tumor infiltrating neoantigen specific T cells, but not that of the peripheral T cells. This framework is applicable for identifying tumor neoantigens or profiling antiviral T cell responses. In addition to using cells in suspension, checkpoint inhibitor studies have looked at tumor microenvironment using imaging mass cytometry. Here in the Nishikawa lab at the National Cancer Center of Japan, they used imaging mass cytometry on the Hyperion imaging system to understand the spatial distribution of PD-1 positive, CD8 positive T cells in the biliary tract or BTC cancer, biliary tract cancer. While disaggregating biliary tract tumor tissue showed that high levels of CD8 PD-1 T cells correlate with smaller tumor size, the spatial relationship of these cells to tumor cells was unknown. Here, as seen in this representative IMC images, PD-1 positive, CD8 positive TILs were found inside the tumor boundary in patients with a high number of this cell type, indicating that patients with early stage BTC may respond to immune checkpoint therapy blockade. Taken together, these findings reveal potential efficacy of immune checkpoint blockades against early stage biliary tract cancer. This slide shows a report from the Annals of Internal Medicine released last week. The authors used an 18-marker imaging mass cytometry panel to enable an unbiased exploratory approach to assess immune infiltration into lung tissue from 2 COVID-19 patients, one who died of cardiac arrest and one who died from pneumonia complications. Differences were noted in the immune patterns of infiltrating CD4 and CD8 T cells, NK cells, and macrophages in a single section of lung tissue from each patient. Recruitment of aberrant CD4RA positive T cells is an immunological feature of COVID-19. Once bacterial pneumonia occurs, Some phagocytes recruited by CD4 T cells begin to play a significant role in lung injury. While not directly related to checkpoint inhibitor studies, and in light of recent events surrounding the pandemic, I included this study to demonstrate the power profile and visualize cell changes that occur during the course of disease, and potentially during or after therapeutic drug treatment. In future studies, additional markers could be added to this panel to examine T cell exhaustion and even provide insight into how drugs poised to treat COVID-19 are efficacious. Thank you for your attention. We'll now be happy to answer your questions. Please enter your questions into the question box and we will try to get to them as many as we can. For those that we can't answer during this webinar, questions will be forwarded to your regional field application specialist for follow-up.
Hi, my name is Wendell Smith. I'll be the moderator of the questions posed today. And I'd like to start with the question that's come in for Hyperion use. The question is, how do you compare the results obtained from different software for IMC analysis? And are they consistent? The answer to that question is difficult because there's never really been a direct comparison between different softwares, whether it's free or the paid models. All the tools have their advantages and limitations, and it's sort of up to you to determine which tool works best for your specific question. I would say that you need to consider first what the question is that you'd like to answer and how the algorithm is designed to work. That's a much more complex answer than, the answer that's much more complex than I'm able to state here. So what I would say is, when you get to the point where you know what the question is, maybe talk to your local FAS for imaging and help you to determine which program to use and which algorithm it utilizes that best address your question with your tissue sample. We have a question here for Andy for Helios. The question is, my dissociated mouse tumor sample barcoding is yielding only 30% and many events are unassigned. What can I do to prevent this?
Excellent question. In order to best help you, I think you need to look at your files and look at your data, and maybe we can recover some of that. Important things to consider are your cell quality. Your viability may be low, and that could be imparting its effects on your ability to recover events. Your viability should be better than that of 85%. Dead cells and debris can soak up, your barcoding and also your antibodies. And if you're not staining at least 1 to 3 million total cells, that could be one of the reasons why you're failing to, achieve the event thresholds you need. Make sure that your cells are thoroughly resuspended prior to fixing cells. This could cut down on some aggregations, give you a better staining properties, and may improve your ability to recover these events and assign them. Best thing for you to do is to reach out to Fluidigm and get in touch with your FAS, and we can do some troubleshooting and probably recover some of your data.
Okay, we have another question for Andy for Helios. Are there alternatives to live cell barcoding besides using CD45?
Yeah, good question. Absolutely. Yeah, you can use a few other markers, and there's a paper available at Hartman et al. It came out recently, and they use CD298 and also beta-2 microglobin. And this will be, it's a surface stain for human cells, so this is for human work. And the advantages of this method is not restricted just to immune cells. So you can, pool stem cells, tumor cells, etc., and use this methodology. If you're doing like a tumor prep, this is something you can use. And so yeah, there are alternatives to using CD45. Again, you could also reach out to your FAS, and we can provide you that reference, or maybe possibly address other markers you want to use.
Okay, it looks like we have one last question. This is for Hyperion. So the question is, I have tissue that is coming in soon and need to know how to proceed if I don't have access to an IMC yet. What do you recommend? A lot of the labs out there are just starting to come back online due to the break that we've all experienced due to COVID-19. And in this particular case, what I would say is if you have access to your antibodies, then I would stain the slide. That slide should be stable for at least 6 months, possibly much longer. Just stored it in the dark at room temperature. Of course, there are other storage conditions that you can explore if you need to go long term or longer than that. But that should buy you enough time for your lab to get back up to speed, giving you the time to process that sample. You shouldn't see very much drop in quality from a fresh, as long as it's fresh stain and it's stained right away. Once it's stained and dried, it should be stable for at least 6 months. We don't have any other questions from the audience at this time. So I'm going to turn it back over to Andy, unless a question comes in the next minute or so. Andy, you can wrap up now, please.
Yes, thank you. From this slide, you can see there's a couple ways to get in touch with your FAS and arrange office hours, phone conversation, Skype demo. And the 2 emails that you can reach out to us at are support.northamerica@fluidigm.com. Across the pond in Europe, you can reach out to eu.support@fluidigm.com. And from there, you will be put in touch with your area's FAS, and they can help address your questions specifically and personally. We also have other upcoming live sessions that you can take advantage of if it's of particular interest. So from what you can see here, we have events, in June and July, and we have everything from top 8 reasons to use mass cytometry, intro to mass cytometry, and of course, introduction to imaging mass cytometry. You can register at fluidigm.com/events. And here's another slide showing some even more live sessions. We have, of course, the FAS Friday Q&A going through August, and we also have deep immune profiling, the MDIPA system that's coming up on June 24th. Again, you can register for these events at fluidigm.com/events. And I'll just bring you back to this slide here if there's no more questions. Remember, support.northamerica@fluidigm.com or eu.support@fluidigm.com. Are there any more questions for today's session? We'll be closing out in the next few minutes.
Nope, I think that's it, Andrew.
All right. Well, thank you very much for your attendance and your attention and your questions today. And again, please feel free to reach out to us at Fluidigm if you need a follow-up question answered. Thank you very much and have a wonderful weekend.